Hello, my name is Devin Lange, and I'm going to be presenting on Loon, a new tool that we developed to visualize large-scale microscopy data using exemplars. The overall motivation for this project is to improve cancer treatment. Cancer is a disease that occurs when cells within your body divide and grow in an uncontrolled way. One of the reasons that cancer is so difficult to treat is because it is highly heterogeneous. There are many different types of cancer, and even within a single cancer tumor, there are different cells could react to the same drug differently. Sort of hand in hand with this, there are many different cancer drugs that already exist to treat cancer, but one of the challenges is actually knowing which of those is going to be effective for a particular patient. And that's the goal that we're going to focus on for this project, is trying to identify which of those drugs is going to be an effective drug. And the way that our collaborators are trying to answer this goal is with a new technology called a quantitative phase microscope. And quantitative phase microscopes can measure how much light slows down as it passes through a certain medium. And it turns out that this can be used to determine the mass of individual cells. Now, in order to use this microscope to test many different drugs at once, our collaborators use something called a multi-well plate, or in this case, a 96-well plate. And here, each one of the circles in this plate is a small self-isolated area where cells can grow and divide, but can't interact with different wells. And this is nice because then you can treat each of the different wells with different drugs or drugs at different concentrations. Here we color the different circles to indicate these different drugs. Now, if we dive down into a specific well, we can see that multiple images are taken at each of these wells. And if we focus in on a single one of those images, this is what we refer to as one imaging location, or sometimes a location for short. And once we have this single image, an important image processing step that you can take is to segment the cells or separate the cell boundaries from their background. Now, this microscope array takes pictures in each of these locations, in each of these wells, and then it automatically goes back to the beginning and starts this process again. As a result, you end up with sequences of images over time, so you can see cells growing and dividing. And with the segmentations that you have, you can find cells that are in a similar position and have similar mass and combine them into tracks. This is the tracking process. And if you continue this over the course of your 5-hour to 5-day experiment, you end up with these tracks of cells. In addition to these images and segmentations and tracks, we record metadata for individual cells, such as the current mass, its shape factor, like how round it is, or any other number of attributes. And with the tracks, you can measure things like the growth rate of an individual cell, or its average mass over some period of time. Now, because this is a new technology with many complicated steps in the process, our collaborators would like a way to analyze this set of images and associated metadata in a way that they can look for comparisons and analyze things, as well as look for problems that they find in the data that are indicative of issues that's in some step in this process. And that's where we came in and developed this tool that we call Loon. And just to give you an overview, I'm going to go over some of the different components of the Loon interface. To begin with, in the top left hand, we have the condition selector. Here we separate the experimental conditions, in this case, the drugs at different concentrations, and display a growth rate curve that can be selected to filter the data to just those conditions. In the same panel, you can switch it to a more detailed growth curve for comparison, where you see those same growth curves separated by experimental conditions on superimposed in a larger area. In the center middle, we have the image selection view. Each of the rows that you see there correspond to a single imaging location, and each one of the little tick marks that you see there correspond to a single image or image frame at that location. Now, once you select one of those tick marks, you can then view that corresponding image within the image view, which shows you that raw image data, as well as the cell boundaries for your segmentation. Here we color code those cell boundaries based on whether that cell has been selected, filtered out, or is currently unselected. And you can do those selections within the cell and track attributes panel. Here we show overall distributions of those cell level attributes like the mass, as well as the track level attributes such as the growth rate of cells. And as I alluded to here, you can see a selection has been made on mass for cells with a mass of 1,000 or greater. Finally, in the bottom right-hand side, we have the exemplar tracks view, which systematically selects exemplar tracks and displays them in this condensed format. Since this is one of the main contributions of our work, I'm going to go into a little bit more detail for how we do this selection and display. So as a reminder, a single track is one cell that's been tracked over time. And our data set consists of many of these tracks in different, exposed to different experimental conditions like the drug types. Again, here this is color. First, we can just separate by those experimental conditions. Then within each condition, we can sort by some attribute. In this case, it's mass, so we see the small cells at the top, large ones at the bottom. But within the interface, a user can switch it to other options dynamically. Once it's sorted, you can systematically select things, select tracks from this list. In this case, the cell at a 5th percentile, the median and 95th percentile. Here again, there are going to be some other options within the interface. But moving forward with this selection, you can then display these tracks and align them strictly by time. And this is something that we did as a first attempt within our tool, but it turns out these tracks can become very, very long for medium to long experiments. And also for our data, since images are taken, at the same location every 5 minutes to maybe every 30 minutes, the cells do not change significantly within the span of one frame or even several. So we decided to do temporal sampling of these cells. So within each track, we can select a set number, here 7 cells evenly distributed. Next, we can also keep track of the time span that track existed. Here I'm showing this with this blue bar above the tracks. Then with these 2 pieces of information, we can realign these tracks to become a lot more condensed while still preserving that temporal context with those blue bars still there above the tracks. And to close the loop, here's how this looks back within the tool Loon itself. We have these 3 actual images with those similar looking blue bars. In addition, we have 2 complementary visualizations. On the far left-hand side, we show a histogram showing the distribution of mass in this case. In general, it will show however you are sampling those tracks. One nice thing about that is with those pins in the far left, you can see where those cell tracks lie on that distribution. Then on the right-hand side, we get a plot of mass over time. And here again, we show the mass over time for the 5th percentile, the median and 95th percentile. Here also, we show the average for that entire experimental condition. So shifting gears here, I would like to do a little bit of a live demo of the tool here. It is also accessible at this URL, loon.ski.utah.edu. Okay, to begin, I'm going to select 3 experimental conditions, these 3 drugs, with a concentration of 20 micromolars. And when I apply this filter, it's going to update these other views to focus in on this data. Next, I want to call to attention within the image selection view, we have these different size red rectangles and these different size small red tick marks. Those sizes are encoding the number of selected cells at those images. So for example, if I collect one of these small images, you can see there's only 1, 2, 3, 4, 5 selected cells at the default selection criteria. And if I want to filter to make this more obvious, I can remove the boundaries on those green cells, so I just see the red ones. And going back to the encoding here, now if I look at one of these large tick marks, you can see there's many cells within the selection criteria. And going back here, there's sort of a medium amount. Next, I want to point out that you can use the keyboard to sort of quickly step between these frames and even use it as a sort of a pseudo way to get some animations here of these images. Next, if you want to update the selection criteria here, you can do that by brushing at the bottom left in these histograms. So here I'm going to brush to get cells with a high growth rate. You can see these 2 red cells have that high growth rate, and these 2 blue ones are not in that really high region. You also saw there that when I hover over this, you get additional information about these cells. Similarly, with the highlighting, we can see now within our exemplar cells, there's actually only one exemplar cell here that is in this high region of growth rate. And if I want to sort of explore the other cells within the same area of the distribution, I can click there in the distribution, and that will pull in a new exemplar for me here. And it actually does that for each of your experimental conditions here. And you can continue adding pins and removing them, or you can move existing pins around and sort of explore the space of this distribution in that way. Similarly, if you see a cell that you find interesting, you can select it to view the surrounding context of that cell. Similarly, now if I select this cell in the image viewer in the top right, it will pull that into my track view, so I can view a summary of its sort of lifetime, as well as help me keep track of it and reference the cell ID for further investigation. So moving on to the discussion here, we think this idea of exemplars has potential to generalize to other areas. And in our opinion, there's sort of 2 criteria that you can think about applying before you can use this idea of exemplars. And the first is that your data should have this rich contextual aspects of it that cannot be summarized. In our case, these are our actual images of the cells. While we can, collect the attributes of the cells, like their mass or shape, that somehow loses aspects of that richer context of the image itself. But you could imagine, of course, as applying to other types of image analysis, but other things that could be similar would be things like network or graph structures, where there again you can measure attributes like the average degree or number of nodes of a graph, but perhaps that full topological structure of the graph is important, and maybe pulling out an exemplar network structure could be interesting. A second criteria we have here is that your data set should be sufficiently large, right? You may have 10 images with rich contextual data, but if you only have 10 of them, you could just look at every single one individually and call it a day. But once you start having thousands of images or tens of thousands or even more, this becomes impractical or really infeasible altogether. And once that's becomes the case, we think that this idea of exemplars of systematically pulling out from that large data set could be useful. So in terms of future work, there's two main paths which we are interested in. One is applying this concept of exemplars to these other situations. So looking at these criteria and considering where else these exemplars could be useful. And the second path is continuing work on the Loon interface. So developing enhancements, and in particular, one area we're interested in is looking at cell lineages, or essentially family histories of cells, and looking at ways that we can visualize that within the tool Loon. And it could be that perhaps there's some middle ground where we do both of these things together, actually. That concludes my talk. Thank you for watching.